In part 5, we are going to start with the environmental factors that influence growth. In the lab, we take into consideration the pH of the media, temperature, and gases present in their atmosphere. Most pathogenic bacteria grow best at a neutral pH between 7.0 and 7.5. Temperature influences the rate of growth, and the optimum temperature must be determined for all bacteria. Psychrophiles grow best at cold temperatures between 10 and 20 degrees Celsius. Mesophiles grow best at moderate temperatures of 20 to 40 degrees Celsius. And thermophiles grow best at 50 to 60 degrees Celsius. Most pathogenic bacteria are mesophiles and grow best at 37 degrees Celsius, which is also the human body temperature. Diagnostic microbiology labs incubate most cultures at 35 degrees Celsius. That's important. Human bacterial pathogens vary in their atmospheric requirements. So that means the amount of oxygen that they need or the amount of carbon dioxide that they need. Obligate aerobes require oxygen for growth. Aerotolerant anaerobes, formerly called facultative aerobes, can survive in the presence of oxygen but do not use it in metabolic processes. Obligate anaerobes cannot grow in the presence of oxygen. A true obligate anaerobe will die as soon as it hits oxygen. Capnophilic organisms, or capnophiles, they grow best in an atmosphere enriched with carbon dioxide for a final carbon dioxide concentration of 5 to 10%. An example of a capnophile is Haemophilus influenza. And those may grow in a candle jar, and that's really exactly what it sounds like. It's a jar that we put our media in that's been inoculated with suspected Haemophilus influenza, and then we put a candle in there. We put the lid on the jar, and the candle goes out when all the oxygen is used up. So if you don't have a CO2 tank, you can try a candle jar, but it's not very effective, honestly. Microaerophilic organisms require a reduced level of oxygen, around 5 to 6%. An example of a microaerophile is a Campylobacter species. Next, we need to talk about bacterial growth. So we talked about the generation or doubling time of E. coli, that it takes about 20 minutes for E. coli to replicate once. So the generation or doubling time is the time it takes for one bacterial cell to divide into two cells. And there are 4 phases of growth. And you'll see this growth curve perhaps on the boards. So during the lag phase, which is the first phase of the graph, this is where bacteria are preparing to divide. And in this media or agar, there has to be enough nutrients in the right conditions, the pH, the right gas, the right atmosphere. So bacteria are preparing to divide in the lag phase. During the log phase, bacteria are dividing and numbers are increasing logarithmically. Then during the stationary phase, the nutrients are very limited, but the bacteria remain constant for a little while. And then the final death phase occurs because the number of dead cells exceed the number of live cells, and this means that all the nutrients are used up and the cells will eventually all die. So this is the classic growth curve, and you need to know all 4 of these for possible board questions. Lag phase, where the bacteria are getting ready to grow. Exponential phase, or the log phase, where bacteria are growing steadily. The stationary phase, and then the death phase. Now, we also need to determine how much bacteria is present in our samples. So how do we do that? We have to determine the cell numbers, and we can do this in 3 ways. We can do a direct count, where we count the number of bacteria present under the microscope. This method is not used in the lab. It would take forever. In a direct counting technique, like counting white blood cells on a hemocytometer, we would have to be able to establish living cells from dead cells, and we don't want to do that. It takes so much time. So the direct plate count and density measurements are used daily in the microlab. So direct plate counts and density. We're going to talk about these in detail. A direct plate count is a way to quantitate bacteria in urines or other liquid samples. The count is determined by using a calibrated 1 microliter loop. Now, you guys, you have seen pipettes and you've seen very small amounts of liquid in your pipette practice, so you know this is a very tiny amount. And we have some 1 microliter calibrated loops that we will use in our laboratory. So in this way, only viable cells are counted, and we count the colonies. So this is a colony. That would be considered 1 colony. And if we use a 1 microliter loop, that means that 1 colony represents 1,000 bacteria. And then we can also use a semi-quantitative plate count for samples that are not liquid. Semi-quantitative results are reported as 1+, 2+, 3+, or few, moderate, many. So keep in mind, when we are doing our cultures, this is an isolated pure colony because there is only 1 colony. It's all by itself, it's isolated, and we are pretty sure it's only growing 1 thing. Now over here, these colonies are not isolated. We can't tell. There might be other bacteria growing, or it could be 2 different kind of bacteria. They're growing so close together we can't see. So we have to grow pure colonies to work with in our microbiology lab. The other count, or the other way to measure cultures or bacteria in the lab, is through density measurements. It's a measure of the cloudiness or turbidity of a bacterial broth culture. This method is used to prepare a standard inoculum for antibiotic susceptibility testing. We make inoculums by removing that isolated pure colony from media and dispersing it in a liquid. We can then measure the turbidity or cloudiness using a spectrophotometer and a Macfarlane standard. So a Macfarlane standard is just it's a cloudy solution, and this is 0.5 Macfarlane standard, so you just compare the cloudiness. We do have a spectrophotometer that we'll be using, or a turbidometer in the lab that we'll be using, and you'll get to do these in the lab, make your own Macfarlane standards. Bacterial metabolism consists of biochemical reactions, and microbiologists use this knowledge of bacterial metabolism to develop diagnostic schemes or flowcharts to analyze unknown microorganisms growing in patient samples. The diagnostic scheme allows us to determine what the unknown organism used for its carbon source, what end products are used, and what produces from substrates is used for metabolism, and whether or not that unknown bacteria produces an acid or alkaline pH in the growth medium. Knowledge of biochemistry and metabolism of bacteria is crucial in the clinical laboratory. So we are going to stop right here and then go into the different ways that bacteria can metabolize and some of the tests that we can use. So this is the end of part 5.